Sounds good. And yeah, and I wanted to say if any of the panelists have anything to add while I'm going through the slides, feel free to share at any time. So I'll be talking first about our advances in rhubarb field management, some of the resources that we have started to develop here, and then some aggregated resources from colleagues in the Northeast. So first, I wanted to introduce the crop. Everyone's here to hear about rhubarb, but we may have different levels of experience with it. Culinary rhubarb is in the species Rheum rhabarbarum, and in the genus Rheum. There are about 60 species in that genus, and it's in the Polygonaceae, which is this family of some crops that you may be familiar with, like buckwheat here, red vein sorrel, other types of sorrel and rumex species, and then also giant knotweed. But rhubarb, or culinary rhubarb, we call it an herbaceous and rhizomatous perennial. So it dies back to the soil level at the end of every growing season. And it's typically clonally propagated because it produces these rhizomes. So when you look underground at a plant, these are the structures that it's producing. And you can divide these and propagate new clones of the original plant. So that's how it's typically propagated. But for culinary rhubarb, we're really interested in the leaf stalk or the petiole. You may be familiar with medicinal rhubarb. That is the rhizomes that are actually dried and ground. But in this case, we're interested in the petioles. And in the northern US, there's a fresh market and a processing industry. There aren't too many acres that are grown currently, just over 1,700. And the top producers are really in the Pacific Northwest, so Oregon and Washington. But it's a cold climate crop. And so across the northern part of the US, there are environments that are favorable for it. So also in Michigan and Minnesota, and then in New York. And there are two ways that rhubarb is produced. You can grow it in the field, as you see in this picture, or in forcing sheds or in hothouses. And these are both in Sumner, Washington, really a capital of rhubarb production in the US. To my knowledge, we don't have many producers in the Northeast who are doing this style, but maybe in Southern Ontario, Canada. And we have found that in the Northeast, there seems to be increasing interest in beverage production with rhubarb. So here's some examples of beverages from New York State that were produced here, ideally with rhubarb that was produced in New York as well. And so we're really interested in identifying traits in the rhubarb that make it well suited for beverage production in the Northeast. So I'll share these recommendations for field management. So types and varieties, soil fertility, some information about planting and field culture, harvesting and storing, and then some pests and diseases. And I wanted to highlight that there are colleagues of ours at other institutions who have created resources for these elements. And so a lot of what I have pulled comes from colleagues in the Northeast. So if you're interested, there are other resources as well for you to turn to. And before I talk about some of the varieties, I wanted to share sort of a disclaimer about identifying varieties of rhubarb. That really identifying cultivars, I think, is at best problematic and almost always unreliable for some of the reasons that I'll share. First, plants are often named from general characteristics or the market appeal of the name, and we rarely know the genetic origin. And there's also, we have found that no keys to identify varieties, and this is probably because the characteristics can change within a year or over years, and so it makes it hard to identify something based on some of the characteristics. This is also difficult because while crown divisions or the rhizome divisions are clonal and they're true to type, seedlings are generally not. And there are producers who collect seed and then germinate those and sell those plants. And I have an example of how this can be a problem. So these are cross sections of the leaf stalks or petioles of seedlings that were grown from seeds that were harvested on a single flowering stalk. So one parent and then produced all of these siblings. And you can see how much diversity there is just in one set of siblings. And so if you were to name a variety based on one of the characteristics of one of these plants, it wouldn't be informative for many of the others. So this can happen when you harvest seeds and propagate them. And there are cases where there are named varieties like German wine, and you have more than one genotype within that name. And so if you just Google German wine, you might see something like this, where someone says that you can get long green stalks with pink splotches. And another result gives you a similar description. But then another nursery sells German wine that is labeled with bright red large stalks. So looking for the name doesn't really tell you something informative. So that can be a difficulty as well. So you can also have cultivars with different names that are the same genotype. So we've kind of inflated the number of varieties that are available, even though they may be the same. So altogether, the best practice that I would recommend and that we would recommend is to rely on descriptions of the characteristics rather than the name when you're searching for rhubarb to purchase. With that being said, I have some examples of varieties that have stable characteristics. If you're interested in expanding your collections and purchasing some. So here are two examples of varieties that are stably green. So they have a green petiole on the outside and in the interior. Riverside Giant, from what I can tell, performs kind of like Victoria, which is a variety that you may be familiar with, and Turkish. Turkish potentially is a different species, but it is reliably green, and these can be found online. Then, if you're interested in speckled or pink petioles, where there's part of the petiole that's green and part is pink, Victoria and Glaskin's Perpetual are some good examples. And Victoria is perhaps one of the most common types that is grown in the Northeast. We have grown it in our trials, and it performs very well here. It's vigorous, and it does have a tendency to flower. And that can be a difficulty when you're trying to manage it. You have to go back into the field many times to remove flowering stalks. Glaskin's Perpetual is said to be similar, but it may have redder petioles at the base than Victoria, but it is also likely to flower. If you're interested in red petioles, and in this case, these are red on the exterior and in the interior, Canada Red and Crimson Red are two varieties that we have grown in our trials in Geneva, and they perform well here. Canada Red is said to be a bit more vigorous than Crimson Red. The petioles can be larger, and it's unlikely to flower. Crimson Red will have skinnier petioles that sometimes seem a little less vigorous, but in both cases, these perform well here, and Crimson Red is unlikely to flower as well. And I wanted to also share these two. Hardy Tardy and Kangaroo are among the very few trademarked varieties that we have come across in rhubarb. These are both made available by Gurney's Seed and Nursery Company based out of Indiana. And they supposedly are based on plants that were collected in Australia, and they market them as being particularly heat tolerant. And maybe Chris or Larry can correct me if I'm wrong. We haven't trialed these at scale, but we have some individuals, some plants of these that we're interested to see how they perform in the summers in upstate New York. So that information maybe a couple of years down the line. So if you're interested in some red petioles, here are some other examples. For soil fertility, I have this table here that I'll leave up. Feel free to take a picture of it if you want. These are recommendations based on soil test results, and they show amounts for nitrogen, phosphorus, and potassium in the year that you're planting and in the years after you plant. So once you have established plantings. For phosphorus and potassium, it's possible to have a sufficient amount in your soils so that you don't need to apply any. But for nitrogen, we're really interested in planting, then having applications of nitrogen in the year that you're planting 2 times, and then once you have an established planting, applying it again. So nitrogen is an important nutrient for rhubarb. So that's something to keep in mind. We mulch all of our rhubarb that we have in Geneva. So here's an example of one of our research plots in Geneva. And this is willow mulch. You can see the shrub willow in the background here. So we have an available source very nearby. So that's what we're using. And this is an example of a commercial planting in Colorado. It's a very different system than what you'd experience in upstate New York. Here they've really mulched kind of the entire field and then planted the plants deeper down into the mulch. And this is to help with water retention, you know, in a drier or arid climate. That becomes important. But that's a good way to add organic matter to your soils and also improve your nutrition. I wanted to talk about divisions. So there are 2 strategies. You can divide the crowns or the rhizomes in the fall, store them over winter, and then transplant in the spring, or you can make divisions in the spring. And there are just some things to think about for making divisions in the fall. Sometimes if you go to a roadside stand, which is shown here, this is in Canandaigua, New York. In the spring, you'll see rhubarb crowns for sale. And what I have found in most cases is that these have been divided in the fall, stored over winter, and then they're being sold in the spring. And that's perfectly fine. We have seen some commercial divisions. In this case, these were made in the fall and then stored over winter with the producer who had made the divisions and then purchased in the spring and shipped. And in many cases, these have secondary contaminants. They'll have fungi growing on them. And it's unlikely that these are really going to cause issues in the production season, but it's hard to be sure that the material is clean that you're working with. So we have made divisions in the spring. And if it's possible, this is what I would recommend doing. You know, in this case, we dug up the entire rhizome. And here we cleaned it. You don't have to do that. This is just for the picture. But in this case, you can see very easily what is healthy and actively growing. And I find that to be helpful right before you need the plants to be growing once you've transplanted them. So I think this is a helpful strategy to know what's diseased, what's clean, and what's actively growing. So I would recommend making the divisions in the spring and then planting. For spacing and plant depth, these are our recommendations. So we would recommend planting at a depth of 1 to 3 inches. This is a picture from a commercial field shortly after a grower had made divisions. And so you can see the depth at which this was planted. And this, we would say, is what we would recommend. So you can see the buds just starting to emerge at the soil surface. And so that's a good target. For spacing, we use a 3 by 6 foot spacing. So we've planted with raised beds, which you can see in this picture in the upper right. This has a drip line down the center, and they're on 6 foot centers. And then we would plant with 3 foot spacing within the row. And so in the bottom picture, you can see those plants are 3 feet apart within a row. And for harvesting and storage, we have some recommendations. Really for harvesting, we would recommend the earliest you'd harvest is in the 2nd year. Ideally, you'd wait for the 3rd year, just so the plants are mature enough to handle that level of harvesting. And you could begin harvesting in May of the year that you're harvesting in. It's important how you harvest. So we recommend twisting and pulling the stalks instead of cutting them at the base. And so in this picture on the right at the bottom, you can see the extra tissue that stays on the base of the leaf stalk if you harvest it correctly. And this helps with the longevity of the petioles in storage, and also prevents there being open tissue on the plants that's wounded for secondary infection or disease to develop. So that's what we would recommend. We'd also recommend to avoid over-harvesting. If you're interested in the longevity of these plants over multiple years, it's important to not remove all of the leaves and the petioles when you harvest. So you'll leave some behind. And our recommendations for yields, sort of a target yield, is 6 tons per acre. That's what's recommended in the Northeast. And we haven't planted at that scale or size, but that's what we would recommend. And for storage, our colleagues have said that they are able to keep their petioles for 2 to 4 weeks in a refrigerator at 40 degrees Fahrenheit or lower with high humidity. So if you're interested in harvesting and storing, that's a target. And there aren't many harvesting aids available for rhubarb, but here's an example from Matt Harvesting. This, I think, was developed for leeks, but was repurposed for rhubarb. And so this is a conveyor belt that's attached to a platform pulled by a tractor, and it pulls the rhubarb stalks up into where you see this gray box. That has a round saw or a blade which cuts the base of the petiole off. So these are likely for processing, and is makes the harvesting more efficient. So if you're interested in really scaling up, there are some equipment available for that. I'll just finish by talking about some of the pests and diseases that we have found in the Northeast. So there's a fungal disease, some viruses, and the rhubarb curculio or the weevil. So the fungal disease we call rhubarb leaf and petiole spot. So it can cause lesions on the leaves, but more importantly on the leaf stalks that you would be selling or consuming. And it's primarily caused by this fungal pathogen called Didymmella rhei. It's been reported across the globe, mostly on rhubarb, but also on a related plant, buckwheat. And it's easy to identify. There are these dry lesions that you usually see on leaves first, but then it can spread to the leaf stalks. And in the infected tissue, the fungal pathogen will produce these structures. They're flask-shaped. They're very, very small. They're hard to see by just naked eye, but in lesions, that's where they develop. And under a microscope, you can see they release thousands of spores. And I have a video to illustrate this. This is at 8 times the normal speed, so it's not happening this quickly naturally. But if you think about in a lesion, you have one of these structures, well, you have thousands of these structures. Every time a rain droplet lands on one of these, or you're overhead irrigating and it lands on one of these, the spores are being released like this. And these can spread in rain splash or high winds. And each spore can germinate and then cause disease on a new plant. So that's how that pathogen spreads. To manage this, we recommend first propagating clean planting material. We think it can overwinter in the underground rhizome. And so every time that you divide a diseased plant, you're potentially spreading new disease where you're going to plant the divisions. We also recommend harvesting the petioles that are symptomatic first so that you reduce the inoculum. So if you see a leaf emerging like this and on the picture here, you can harvest that first and reduce the chance that that will spread inoculum in the growing season. We haven't identified any resistant cultivars yet. That's something that we're interested in doing. So we don't have any recommendations yet, but stay tuned. And there are some fungicides that are recommended in the Northeast. We have not trialed these ourselves, but we don't have any reason to think that they wouldn't work. What I wanted to highlight is that there are different frac groups available that are recommended and that our colleagues have tried in other states in the Northeast. So those are some examples. Next, viruses. So there are several viruses that have been reported on rhubarb. And in many cases, the symptoms are hard to distinguish from one another. You may see something like in this upper image here, and that's caused by tobacco streak virus. In this lower picture, we don't know the cause of those symptoms, but that may lead you to think that there are viruses in that tissue. And you'll notice that many of the names of these viruses are associated with other hosts, like cherry leaf roll, cucumber mosaic, strawberry latent ring spot. So in these cases, these are viruses that were named for causing disease on that original host and have potentially jumped to the rhubarb as a new host or evolved to infect rhubarb. We have found some of those viruses in our own collection of cultivars. So we first we saw these symptoms in the bottom picture. These are severe symptoms that we saw on a single accession of ours, a cultivar, and we were able to macerate the leaves that were symptomatic and spread the sap of that onto new healthy seedlings and cause new symptoms. And so the picture you're seeing at the top, that's an infected plant. So we spread the virus and demonstrated that that can be done. What I've underlined here are the viruses that we've found for the first time in rhubarb. So these have not been reported before, to the best of our knowledge. Beet curly top, broadleaf dock virus A, and tomato ring spot virus. And I say here that we've confirmed tomato ring spot virus because we were able to reproduce symptoms, which you see in the pictures on the right. And lastly, managing viruses. There are not a lot of recommendations that are all that helpful, but first you can test your soil for nematode vectors before you plant. So a number of those viruses are transmitted by this nematode here, the dagger nematode. And that is hard to manage once it's in your soil, but would be helpful to know if it's there. What you really should do is dig up and destroy symptomatic plants. So if you see those symptoms, you can't cure it in a plant, so it's best to remove it if possible. And also, if you have adjacent weeds, especially ones that are closely related to rhubarb, like species of rumex, like you see here, to remove and destroy those. Because we have reason to believe that those can be a host to some of those viruses as well. So the last pest that I wanted to talk about is rhubarb curculio, or the rhubarb weevil. This is a relative of the cotton boll weevil, which some of you may be familiar with. This is native to North America, and this is important to consider because rhubarb is not. Rhubarb is native to Asia, and the rhubarb weevil originally had these host plants. So dock, which is a relative of rhubarb, and thistle and sunflower. And this is how it evolved with these plants as its hosts. And that's interesting for the life cycle because the adults, which you can see in the bottom pictures here, they emerge in the spring. We've seen them in early May. They overwinter as adults. And then they will deposit their eggs in the leaf stalks of their hosts. So dock, thistle, and sunflower, but also rhubarb. And you can see the damage that they cause in these pictures on the right, where they've deposited their eggs. And after 7 to 10 days, the eggs will hatch in all of the other host plants, but rhubarb. So even though they can't reproduce on rhubarb as a host, they still will lay their eggs in it, which I think is an interesting example of a plant being introduced to North America and being related to a host of the insect, but they didn't evolve together. And so it's a little frustrating that you just wish that the rhubarb weevil would evolve quickly to realize that it's causing damage, but of no benefit to itself. So anyway, that's an interesting problem. Really, to manage it, what we would recommend is removing other hosts that are nearby, so dock, thistle, and sunflower, and killing the weevil when you come across it. And with that, I would like to thank everyone who's involved with the project. And I can, well, I guess if there are questions in the chat that make sense to answer now, I'm happy to do that.
There are a few questions. I'll just repeat them so then you can answer them live.
Okay.
Can you still eat rhubarb once it has flowered?
Yes. So eating rhubarb once it's flowered is totally fine. Really, the problem with flowering is that it is an additional stress to the plant and it can reduce its productivity when it's flowering. But there's no problem with eating the leaf stalk if there is a flowering stalk at the same time.
Okay. How soon can you sustainably harvest again? If I harvested on Monday, can I harvest more if it's ready on Wednesday, the following week? How far into summer or fall? Those.
Yeah, so I think it depends on how heavily you harvested the first time. Um, we don't find that leaves emerge that quickly within a week, but if you've harvested just one petiole at the beginning of the week and you want to harvest another one from the same plant at the end of the week, that's totally fine, provided that you're leaving enough leaves on that plant throughout the growing season. We have harvested late into the summer. Sometimes there's a second sort of flush of foliage into August or so. And that's perfectly fine to harvest then, again, provided that you're leaving leaves for the plant. But we think that the quality of the petioles can be reduced towards the end of the growing season. It's anecdotal, really. We haven't done that research, but if you enjoy the petioles in August, then that's perfectly fine.
Okay, and then proper disposal of contaminated vegetation. Should it be composted or thrown in the trash?
So if you are confident that you have a disease, and you're throwing away that material, it's best to not have a cull pile that's right next to your planting. I think it would be fine to compost, provided that you're not going to take the soil from your compost pile and put it directly on the rhubarb. This is not at least the fungal pathogen isn't so severe that it's going to decimate your garden. And we don't think that it will go to other garden crops. But yeah, so, don't use the diseased leaves as mulch on your rhubarb, but you can probably compost it.
Okay. Are slugs a problem as well, or no?
Larry and Chris, correct me if I'm wrong. We don't really see a problem with that. We've had caterpillars eating the foliage, but not the leaf stalks. And I don't believe that I've seen slugs. I have seen snails eating the leaves, but not the petioles.
Okay.
I've seen slugs in some rhubarb plantings
Okay.
that particularly if they're like heavily mulched.
Okay.
And Robert, were they attacking the petioles as well?
Yes.
The leaf? Yeah.
Oh, okay. That's good to know.
Sometimes the early, as soon as you have a generation of that are hatching, you go out there at night and there'll be like hundreds of little tiny slugs all over it. Eventually, they can feed on the petioles. They do go up into the leaves, and then eventually some will get big enough, and there'll be more visible damage. But
Mhm.
and it's periodic. I mean, it doesn't happen every season. Haven't really figured out the correlation, but spring, early summer seems to be, maybe it's wet years. I don't know.
Um, the next question is, will forcing rhubarb change the petiole color within the types? Like forcing green petiole varieties change color to pink, for example?
Yeah, that's a great question. We have not ever tried forcing rhubarb at our research station. So I'm really, that is out of my
Well,
wheelhouse.
I guess sort of we have, but we haven't forced it outdoors, but we've dug up rhizomes and brought them into the greenhouse and had them grow in the greenhouse.
Right. I
And
guess yeah, if you're talking about in with exposure to sunlight versus in the dark, I guess, yeah, we've certainly grown them in the greenhouse. That is true. And we haven't
they're
seen,
true to type.
yeah.
Yeah.
Yeah, that doesn't seem to change anything. I have heard reports of certain varieties performing poorly in when they're forced in the dark. And I don't know if that's a change in color or maybe it could be due to the size of the rhizome that was planted. I think that's a really interesting area of research that we haven't explored. But if you do it in the greenhouse, we don't see any changes.
Okay. We have lots and lots of questions keep rolling in. Somebody's not having luck with the rhubarb. Does it need more full sun or more shade? Looks like your plantings are in full sun, but how would you weigh in on that?
So yeah, our plantings are in full sun. We have heard of people having rhubarb in the shade that performs fine, maybe part shade. Yeah, it would be hard to know what to improve without seeing this person's setup. But I think Chris and Larry, you have quite a few plants that are in partial shade and they're doing fine. Is that right?
Yeah, and in our research field, in the heat of August, the rhubarb really doesn't do well in the full sun. And I think in that scenario, it would actually do better if we had partial shade. And rhubarb is also, even though it grows in all of our backyards, it's kind of fussy about water. It doesn't like too much water. If it sits in water, it becomes very unhappy. But if it gets too dry, it really it's very sensitive to extreme dry. It has a fairly decent rhizome and root system, so it takes it a little while before it gets droughted. But once it gets to that droughted stage, it becomes fairly unhappy. So even watering seems to be what it likes the most, and it likes the sun, but not, I would say, not extreme heat.
Okay, thank you. So the next question is, should the flowers be cut back or allowed to go to seed on new plants?
So on new plants, if it's in the first couple of years of planting, I think we would recommend cutting the flowering stalks off. Really, that is mostly of value to you if you want to collect the seeds, and if you don't, then there's go to seed. So we cut them off even if they're mature plants. We're just not interested in having that many seeds and having it reduce the productivity of the plant.
Okay. Is there research on yield effects of disflowering?
So if the yield decreases if they're flowering, is that the interpretation?
Yes.
Yeah. To my knowledge, there haven't been yield studies and comparisons done, but overwhelmingly, that's what people say. We have seen our plants die back earlier in the early fall if they flowered. And so that would certainly affect your yield if you're going to harvest in the fall. You can't do that. And it's possible that it stores less energy over the winter because it wasn't, you know, photosynthesizing for as long in that growing season.
Okay, how long do you keep the original plant? How long is the lifespan of one plant?
That's a great question. I don't think we have a definitive answer to that. There are people who have reported plants in their backyard that have been there for over 20
Decades.
years. Yeah, you know, yeah, decades. I've read reports of people saying upwards of 100 plus years. If you're harvesting every season, I think it would be hard to keep a plant going for that long. But they're really, really long-lived perennials.
Very good. Another question about slugs. What would the control methods be if you do have a big slug year?
Yeah,
I would
Robert,
throw
do you-
that to Robert.
yeah.
Oh, Robert.
Right now, I mean, it's just the choices aren't very large
Which
for
to be
that.
fair, that's why I threw it to Robert.
Thank you. It's some of the slug baits that as long as you keep them on the ground, they're seem just about as effective as anything else. And I'll have to double check, but I think there was, or maybe still is, an organically approved one as well. I know I've actually been sitting in on a slug and snail working group. And there's a lot of research looking to be completed, looking at nematodes and other biocontrols for slugs and snails. So that's something that I'm trying to keep up with to see if that might, once there's something that becomes definitive that might be able to be used, we can try that on rhubarb as well as other crops. So we'll just have to see.
Jacob, at some point, do you want us to cut and talk briefly about the juicing since Chris Gerling is on, or would
Yeah.
you rather talk about these horticultural questions?
No, we should probably take a break, questions, and talk about if Chris is ready to talk about some taste and juicing. Because I think that's really interesting and people will definitely want to hear about it.
Hi, everyone. Hi, Jacob. Yeah. Yeah, I mean, so we've done some work based on.
Wait, Chris,
Oh.
to be fair, we did not introduce you before you got here.
Oh,
So
sure.
go ahead and introduce
Sorry.
yourself.
My name is Chris Gerling. I am a senior extension associate in the Department of Food Science at Cornell Agritech. I was just at a wine tasting that I had to come straight from no one feels sorry for me, but that's what I was doing. So sorry about that. And yeah, and so I've been working with Libby and Chris and everybody to take some of the rhubarb that they are growing to do some processing and to do some we've made like basically a wine with it to look at some of the fermentation capacity and look at some of the chemistry and see where it how it works and how it goes, because we know that as Libby showed, there's a number of beverages that people have produced and are producing both here and in the UK and in places like that. So we wanted to kind of just get some basic numbers to go with people's experiences, because as we know, rhubarb is this amazingly traditional thing that everybody knows about, but nobody actually knows about, right? Is the way it goes. You know, we have all this experience, but we don't have a lot of data. And so anyway, so we had 4 different lots that we used this last season. And so, you know, there's kind of 2 factors. There's kind of the processing aspect of it, and then there's the chemistry we get from the product afterwards. The processing part is something that we got some advice from people who had been using it in juices. And so, you know, it had to do with how to chop it and then to freeze it, freezing it and then thawing it or for pressing is one of the major strategies that we learned about for increasing yields of juices and making and the pressability of it. We have and there are machines for chopping out there, not usually like made for this, but there are some machines that either people have built or can be customized from other products, as Libby was showing you with the harvester, adapting and adopting other technology, because it is something that can be labor intensive if you don't have a way to do that. And so that's one side of it, and that's something I'll be interested to hear what people are doing and kind of your experience and your challenges along those lines. Then once we had the juice, we made some basic, I call them wines. Rhubarb is a product that people often blend with other products with, make a rhubarb cider or rhubarb beer, things like that. And it lends itself to that particularly well. But we were just doing standalone for kind of some proof of concept and to get some reliable data about the chemistry of the product and how it, just kind of behaved and looked. And so what we found is that, the instincts to blend it is particularly apt or good, because the one thing it's not a source of is sugar, right? So that's fermentable sugar is where it kind of lacks. The range that I'm looking at for this data, we were like 3.5 to 4.5 degrees Brix from our measurements, which translates roughly to percent fermentable sugar. It's a soluble solids measurement. It's weight per weight, but 4. So and when we ferment, we get, we figure we get roughly half, maybe 60% of that sugar becomes alcohol. And so that's not a very high alcohol yield if you're making a product that's going to be a standalone drink. If you're trying to compete with beer, wine, cider, things like that. So we chaptalized, which is we added sugar so that we could ferment to a more traditional alcohol volume. We went up to 18 degrees Brix, which gave us roughly 11% alcohol. And but what we found with the other chemistry was fairly good in terms of the pH, in terms of the amount of acid, in terms of the amount of nitrogen. The other things we look at when we're looking at grapes or apples for fermentation, you want a certain amount of acid for both flavor, but for also microbial stability. So because if the pH is too high, then you have all sorts of risks of spoilage and things like that. And the pH of this was actually a pretty good range for what we see for a lot of wine that we're making. And so that was good. The acid, you all may know, is mostly it's malic acid. So it's the acid that we traditionally find in apples, as opposed to like tartaric acid, which is what we find in grapes, which is once again fine. That's what that's what when we're making cider or apple wine. And there's plenty of that, maybe a little much actually, but it's it's good. It's in the range of we were looking at like 10 to 15 grams per liter, which is on the high side for wine. If you're trying to compare to like wine, it's not out of the range, same order of magnitude, it's just a little higher. So we could work with that. And then there was also plenty of fermentable nitrogen, which was kind of a nice surprise. It's not, it's something you can add if you're making some products, but that's something you don't have to add because the rhubarb had it. And so it actually works pretty well in terms of a fermentation situation, because a lot of the chemistry is pretty similar to these fruits that we're usually making these products from. And so we were pretty pleasantly surprised with that. We got good color from some cultivars. The colors varied. I have a picture I can dig up. Libby might have the picture too of, we have the products, and some of them had a nice red color, and they held on to the color. The color was stable. We've heard people say that they worry about the color being stable, but the nice red rhubarb color, at least some of these cultivars, was nice and was stable. And yeah, it does vary depending on the cultivars, at least what we saw. We don't know if that's a seasonal thing or if it's a cultivar thing. Thank you, Libby. I knew you would or Chris or whoever's doing it. Yes. Yeah. And so yeah, we could see these different different colors, and it as you can see, it kind of relates to the color of the plant, or the the product as we bring it in. So yeah, we were happy with that too. For me, what I want to know from producers of both the product and the beverage, is starting to think about the scale of that we're going to be talking about, and how much people are going to need, and how much people can supply, and what's a reasonable, when we start to think about not just proof of concept, but tons of this, how to both make all that feasible. I think that's the next frontier for us. But in terms of the chemistry and just the physiology and the product itself, and it lends itself pretty well to this. And we should also mention that we tasted it, of course, and we were nice taste. There's some was more intense and more of what we might think of as rhubarb. Some was less, but it was definitely pleasant on its own, and there's nothing worrisome about anything that we actually that we tasted. Varying levels of intensity again, and something that could be blended more or less, but all that is to say it seems perfectly promising from in principle, and then I think logistics is really the next challenge for us. So yeah.
Thanks, Chris. The one question I have, maybe you answered it kind of. Libby kind of talked about plants with high acidity and plants with like lower moderate acidity. What does that really mean in overall flavor profile of a plant with high or low acidity?
Well, from my perspective, so we have, so we had the pH of them is fairly similar, and the pH we're not so worried about. But when we look at the acidity from a grams per liter perspective, from a titratable acidity perspective, we see, I maybe 10 to 15 grams per liter of malic acid. And so that's going to relate directly to how sour you perceive it to taste. And so, 15 grams per liter or whatever, 14, that's pretty high. That's pretty sour if you're just going to have that product, straight up. But of course, the counter to that is lemonade, right? Lemon juice is very, very sour. We add sugar until it becomes less sour. Everyone can pick their point, at which point, sometimes it's not perceptibly sweet anymore. It's just less sour. So sugar is something we can obviously use to moderate that sourness, aside from blending with other products that might have different characteristics. But yeah, I mean, it's a lot of acid. It's plenty of acid, but there's ways to get around that. So yeah.
Let's see. So more acid equals more sugar and strawberries in my pie.
Exactly. Yes.
Perfect.
Yes.
Awesome. Chris, I see and Libby, I see you are answering some questions in the chat. Thank you.
Yeah, I can talk just a bit about the experiment that we're doing this year. So we have in our research farm, we have a planting that has like all of the diversity of rhubarb that we have been able to procure so far, whether we've purchased that or gotten it from a germplasm repository, sort of the USDA bank of rhubarb. Some of those have not survived our winter, which is good to know. Not everything is super, super winter hardy, and not everything will do well in places that are hotter. And so we have about somewhere between 40 and 50, I believe, cultivars in that plot. And then we have another field that has about 100 plants, each of 4 different cultivars. That would be Victoria, Crimson Red, Canada Red, and McDonald. And those are the ones that we've been giving to Chris to test. And then this year, we harvested the seed of two different cultivars. One was a German wine plant, so we want to see if those green stems, when you have the seed from that plant, if all of them will turn out green and what they look like, and what that looks like if we actually try to juice it. It's at least the plant that we collected the seed from is really, really tart. Like, I love rhubarb and I'm happy to grab a stalk of rhubarb and eat it. That was a hard one to eat. And then the other plant that we have 500, we'll be planting 500 seed in the field from is like sort of our, I think it's our, well, it's certainly my personal favorite. I'm going to go ahead and call it the Cornell Rhubarb's team personal favorite. It has what I call, it has that really nice rhubarb flavor, which Chris Gerling is going to help us figure out. I call it oomph. Right? There's a flavor to rhubarb that I think nothing else has. And that particular variety, which to be fair, is not yet named, and the seedlings are not yet named because it came from my mother's house and my grandmother's house and moved across the country with me. But it'll be interesting to see what those 500 plants look like. And I think sometime in June, Jacob, we're going to have a field day. And if anyone's near Geneva at that time, you can come. And what I want is for people to walk through the field and look at the 500 plants of each of these different, siblings, progeny, babies from the original plant, see the differences. And then perhaps we can have a taste test. And I would love to get some feedback on the flavors that different people get when tasting different types of rhubarb. Which I think would be really great. But I think it will be interesting to see what the differences are between these the seeds that come up. And I was answering a question. Sandy McDonald sort of asked that same question of what's the vigor of a plant from a seedling compared to from a division. And when we have always started seedlings in the greenhouse and then transplanted them to the field. And when we have done that, those plants have done very well. I would say they've done just as well as divisions that we have purchased. Libby, what is your thought on that?
Yeah, I would agree. Yeah. I don't think you can tell a difference, really. Yeah.
No, by July of that first season, everything looks the same, and certainly by the second season. I will say that the rhizome, the picture of the rhizome that Libby showed, that was huge. That was a plant that, that was a 3-year-old plant. Yeah, so the underground portion of a rhubarb plant gets very big very quickly. We also add compost. I'll say we try to add compost both roughly now, sometime between now and end of the first week of April. And then we hit the rhubarb with compost again in late June, early July. And when we do that, many of the plants will then throw that second flush of really nice leaves and petioles. One thing I think have not done is compared the flavor and acid characteristics of earlier season and later season stalks, which is one of the things we would love to do with Chris Gerling.
Thank you, Chris. There are still a lot of questions in the chat. We're coming up on an hour. So if people have to leave, that is okay. But I do want to just answer these questions live, and then I can kind of create a resource document later that might have some of these. So did you just answer that question? Yes.
Yes, about the seeds and the divisions. Yes.
Yep. Okay, then we'll go on companion plantings for better production or pest management. That feels like a good question. Libby or Chris, whoever wants to take it, I guess.
Robert?
Robert.
That's a great question. Yeah, we don't know. I don't know of anyone trying any combinations of things. Chris, I don't know if you're aware. But yeah, certainly you wouldn't want some of the host plants to the rhubarb weevil right next to the rhubarb. That would be like an antagonism of companionship. But yeah, I'm not aware of any beneficial companions.
I will say that for the rhubarb weevil, we're working with an entomologist who is using scent traps to lure the cucurleos out of the rhubarb field and into that trap, similar to, say, a Japanese beetle trap.
All right. Here's a question, and it how can we identify cultivars from backyard plants? And is there any way to increase sweetness in backyard plants with more sun, harvesting? Sounds like a big research question. Yeah. But have you found any way to increase sweetness in your plants, or just depends on the cultivar, really?
I haven't seen any way to increase sweetness, and that's one of the things that I'd be really interested in seeing is if you have something in shade or part shade compared to sun, do you see differences either in color or in juice composition? You're right, Jacob, a great research question.
Great. Yeah, lots of great research questions coming out today. This looks like our last question. It's a little long. Are you trying to maintain a deep organic layer around the crowns, or are you keeping the compost back in volcanoes or worked into the soil adjacent to the crowns? A lot of opinions on both sides around deep mulching and organic amendments. Concerned about trapping or encouraging moisture crowns. But what are your thoughts on that and the mulching and the crown and compost?
I think all of those things are correct. What we do is in the spring, we just throw it over the top. We
Okay.
don't work it in. And then in late June or early July, we band it on either side of the plant. So we don't work it in. My guess, and I'm throwing a question to Robert to some extent though, is that with that extra organic matter, if it is a wet year, we might be increasing our chances of things like snails and slugs. Yeah.
Yeah,
But-
yeah, I mean, it could be.
But that's what we do. And in part, we have a lot of plants and it's quick, right? It's quicker to just toss the compost on. We have a bucket loader and we just shovel it out of the bucket as we're going down the road.
Okay. So very easy for like a backyard person, something that's similar to like asparagus or your backyard hop plants that I do.
Yep, exactly. Yeah, and at our house, we do the same exact thing for hops and rhubarb.
Yep. Okay. I think that is all the questions we have. Just a few comments from the survey. Most of the people here have some experience with rhubarb. Most have experience with cooking, and some have plants, but they find they struggle a little bit. And a lot have successfully grown and divided rhubarb. So a lot of good representation here. And so now I have some questions to make some future resources and documents for people. But yeah, so we'll connect again. We'll connect after this webinar. I will, are there any more thoughts to share with our, we have a raised hand.
Yeah, Jacob, I was gonna thank you for organizing this. We really appreciate it. And say that we do have producers that contact, all four of us saying, do you guys know of a source of rhubarb? So if anyone out there has a fairly large size rhubarb patch and is looking for someone to sell, I think that's information that people that are trying to source large amounts of rhubarb, they don't know where to go. So it would be great to turn rhubarb into more of a commodity crop. Robert?
It's true. I've got, been contacted, somebody who wants to buy larger quantities for craft beverage. So they've got money to spend on it. So yeah, it's just looking for sources, consistent sources, and looking, to see. And I think just from the past experience, I think that